difference between meiosis and what? And mitosis. Altogether. Remember, we have meiosis 1 and meiosis 2. And we have what now? Mitosis. Again. Now, what is meiosis? Meiosis is another type of a cell, what? Division, whereby diploid cells divide to form 4 dissimilar haploid cells. We have meiosis 1 and meiosis what? 2. Again. Now, look at this diagram. Again. We say, number 1, we are going to have a structure like this. Again. Let's put chromosomes inside. Let's put 46 chromosomes. Again. Remember, 46 chromosomes, these are full complete set of what? Of chromosomes. Which means when how does it first like that? You are a diploid. Already you are a combination of how many people? 2. You are a combination of a father and the what? And the mother. Altogether. Both of them have donated what? 23 and what? And 23. To form a what? A 46. Which means when you are a what? You are a diploid. Altogether. Or you can say you are a 2N. Altogether. Now, meiosis 1 is going to what? Is going to double the number of what? Of nucleus. The nucleus number is doubled again, but the number of chromosomes is halved. Which means it's going to be 46 and then it comes to 23. This side divided by 23. Altogether. We have how many cells now? 2 cells. That is meiosis what, guys? 1. Altogether. Meiosis 2, we are going to have something like this. Altogether. Meiosis 2, remember guys, the number of chromosomes was halved in what? In meiosis 1. The number of chromosomes was what? Was halved. Altogether. And then the number of nucleus, the nucleus was what? Was doubled. Nearly 1 again nucleus. Now it's 2. But the number of chromosomes is what? Is halved. And then we also have what, guys? Crossing over. So how many things are happening in meiosis 1, guys? 3. The first one is what? Chromosome number is what? Is halved. The number of nucleus is doubled. Then now there is what now? Crossing over. Which is what, guys? Exchange of genetic material. Happening at which phase? Prophase 1. Prophase 1? Exchange of what? Genetic material. Which is called what? Crossing over. But meiosis 2 now. What is happening in meiosis 2, guys? The number of chromosomes remain the what there? The same. Which means it was 23, that side again is also going to be 23. Which means the number of chromosomes remain the what there? The same. Altogether. But the number of nucleus, it was 2 this side again. Now they are what? They are 4. Which means the number of nucleus is still what, guys? Doubled. Do you see? It is doubled from what? From 2 to 4. Altogether. Is there crossing over meiosis 2? No. Crossing over has already taken what? Place. It takes place only at meiosis 1. Which means meiosis 2, there is no crossing over. Because crossing over has already taken place at meiosis what? 1. Altogether. That's why here we are saying another type, which means yes, it is saying well, there is someone else who is also what? Another type. Altogether. Of a cell what? Division. The cells were dividing now. Altogether. And then diploid. This is a diploid again. And then it divided to form what, guys? 4. This is 4. 1, 2, 3, 4. Like actually one, guys. Whereby we are getting one characteristic to get how many combinations? 4 on a mono hybrid. Altogether. Now we go to mitosis. What is mitosis, guys? Another type of a cell division whereby identical daughter cells are formed. Means the parent and the offspring are what are the same in what? In number. When I look at them, I cannot identify who is the parent, who is the child altogether, because the number of chromosomes are what? Are identical. Which means I can have a parent having 7 chromosomes, for example. It can be 7, it can be any number, it doesn't matter, guys, altogether. Even if it can be 9, the parent can have 9. The child are going to also have what, guys? 9. Because we say identical daughter cells are going to be formed out together. But physically on our body, how does mitosis help us? Remember, double R. Again, the first R is for repair. The second R is for replace. It repair and replaces what, guys? Damaged cells, tissues, or organs. Like replacing a kidney, again, and then repairing my skin when I'm hurt. How together, guys? We're using what now? Mitosis. But don't also forget, guys, you guys, when you are doing development of what? Of an embryo, we use also what, guys? Mitosis. Do you remember, guys, when you're developing an embryo, would you what? Remember, we have what? A zygote. Again, after zygote, we have what? Morula. After morula, we have what? A blastula or a blastocyst. Altogether. After blastocyst, we have what now? Embryo. Altogether. After embryo, we have what now? A fetus. A fetus. Again. Guys, how do we get a zygote? We get it through what? Through fertilization. Altogether. How do we get a morula? How do you get a morula? Through what? Through mitosis. Altogether. And then how do we get a blastula? Through what? Through implantation. So this is the most important thing. We fertilize. We fertilize a sperm cell and an egg cell to get a what? A zygote. After getting a zygote, the shape will be like this, guys. Altogether. And then we go to what? A morula. Identical daughter cells are going to be formed. Again, which means this is going to be our cell. Now it's having identical what? Daughter cells. This is now a morula. But a blastula or a blastocyst implantation is planting altogether. Which means one of them is going to develop. The rest are going to serve as what is nutrients. Which means it should be like something like this. And then one of them, it develops. And then this is what now? Implantation. Out together. And we know exactly or an embryo. Again. That's an embryo. That's an embryo. And then a fetus usually as long as I should be fat.
Where are you fat?
Was mana hata kile tuanza pale kwa kutoka kwa wake. Hakapata mafela. Cesarean section au kata. Mwana sheba fatsi kutoka kwa pele maheta au ulahani. Again guys, we push. Mara hakapata mafela wale. Sakio. But here guys, this one I was just showing you that you guys also use what now? Mitosis. When you are doing what? A morula. Out together. Sharp. And then last but not least, guys, we need to understand that there is the 2 marks when you're doing a genetic cross again, whereby you're going to say what? P1 phenotype. What is the phenotype? A physical appearance. P1 genotype. What is the genotype? A genetic composition. Or something which cannot be what? Cannot be seen. Out together. Like myself, I'm a yellow bone inside. And then meiosis, we talked about it. Here it is again. And then gametes. Gametes are sex cells. F1, I mean fertilization. That is a fusion again. We are fusing what? An egg cell and a sperm cell again. And then F1, the first feature of the what of the genotype. And then F1 phenotype. And then the last question can also be ratio, percentage, or what? Or probability. Are we together on that one, guys? Yes. Now let's do only one cross again. Now, let's say for example, yellow plant is dominant over what? Over white plant. All together. Yellow is dominant over what? Over white plant. All together. Now we are going to have what now? A yellow plant. We are going to cross it with the what? With a white plant. Remember, we said yellow is what is dominant over what? Over white. Again, And then they're going to say to you, cross a heterozygous yellow plant and the what ended? And the homozygous what? White plant. Obviously, they're going to give you keys again, guys. They're going to say, for example, capital letter Y for what? For yellow. And then small letter Y for what? For white. Out together. Now I'm going to have what? I said heterozygous what? Yellow. Which means it's a capital letter Y and what? And the small letter. This side I said what? Homozygous what? White. Which means these are going to be two what guys? Small letters. Out together. Now when I cross them, remember I need to do meiosis. We said it's a what? It's a cell division. I'm going to divide these things and put a capital letter this side and then a what? A small letter this side. Which means this is what I'm going to have now. A capital and a small letter. You can even put something here to show what? Division. You still have your small x as a crossing again. I'm going to divide a small letter this side and then a small letter this side. And then I put something like this here. All together. Now I'm going to cross. Automatically, I'm going to have my formula, which is called the J what? JF. I get, which is the John's formula. I get sharp. Yeah, formula you. But a lot of teachers, they know it all together. Sharp. Okay. Now in your head, guys, just have 1, 2, 3, 4. All together. Now I'm going to say 1 is to what? Is to 3. 1 is to
4.
2 is to
3.
2 is to
4.
Again. Now when you did this now, you have your formula. All together. Now I'm going to drop 1, which is the capital what? Letter. And then I'm going to drop what? 3, which is the what? The small letter. Which means I'm done with the 1 is to 3. I go to the second one. 1 is to what? Is to 4. Which means I'm going to drop the capital what? Letter. I'm going to also drop the what then? The small letter. Which means I'm done with the 1 is to 4. All together. Now I go to 2 is to 3. Now I'm going to drop 2 is to 3. I have a small letter. I have also what? A small letter. I am done with 2 is to what? Is to 3. That is what I have altogether. I go to the last one. 2 is to what? Is to 4, which is this one. 2 is to 4. I drop the small letter. I also drop the other, the small letter. Now I am done with this altogether. Now we look at the last part, the phenotype. The F1 what? Phenotype. You look at only the first letters altogether. The first letter is capital again. The first letter is also what? Capital. How many capitals do I have? 2. Which means I'm going to have what guys? 2. 2 which are what guys? Look at them. They are yellow. Altogether guys. 2 which are going to be yellow. Yellow what? Yellow plant. And then small letters. Again, means I'm going to have 2 which are what now? Are white. 2 white what? Plants. I want you to know that one. Yes. And then from there, we look at the last part now, whereby we are going to talk about the types of what? Of dominance. Again, how many types of dominance do we have? 3 types of dominance. Again, yes. The first type is going to be what? To be complete dominance. Again, the second dominance, incomplete dominance. Again, the last dominance is what is a co-dominance. Now let's look at this. If we say something is a complete dominance, if we're talking about color or height, again, if it's color, if all of them are black, it means it is going to be what now? 100 what? Percent. Again, it's 100 what? 100% black, which means it's a complete what? Dominance. If the class is consisting of everyone as a female, it's 100% what? Females. But incomplete, it will always result to a 25% is to 75%. Out of the 4 kids, you have 3 which are black, 1 which is white. This one is questionable again. Again, but this one is going to be 1 is to 3. 3 are black, one is what is white altogether. And then a codominance is going to be 50-50 again.
Yes.
Juliet Amujulung again. 50-50 altogether. We go to town, Richard results are 1,000. We don't have 500, yeah? We have 500. But we again. And then it's a codominance about 2 is to what is 2? 2 altogether. But guys, you're also allowed. How come 1 is 2? 1. It's also allowed altogether. It's the same thing with 2 is to 2 kappa 1 is 2 is to 1. Right again, guys. Sharp way. From here